It's my pleasure to be here. First of all, thanks to the organizers for inviting me, especially if Timios helping me figure out what exactly is expected out of this talk. And actually, I'm glad that I went third after two speakers because I feel like we are on the same page and in terms of what the regulatory expectations are and what the industry expectations are. And hopefully, this workshop will also help us understand, as you may or may not know, FDA pharmacometrics is also going through some transition. So it will also help us focus on what our future needs are. And I'm really glad to be here. So what I plan to do for this particular talk is to highlight the growth of pharmacometrics. That's to see how we have progressed over the last decade, to put it in perspective as to what the scope has changed, where uh, we are focusing our energy, what is our major focus areas are, and after that, I'll give you two very high-level examples on the impact. And the reason for these particular examples is not about the technical details of it, but as a concept that we are driving at FDA in terms of using quantitative science. So I'll begin with the definition of pharmacometrics, or you heard modeling and simulation, quantitative analysis. The term has been used and thrown around in different contexts. But the way, at least we view pharmacometrics, the definition is the science of quantifying disease, drug, and trial characteristics with the goal to influence drug development, regulatory, and therapeutic decision. And I want to make a note here that in this definition, you'll not find a word model or simulation. And I think we are very particular about this because it is not about model, it is not about the technical details as to what decisions we are influencing and what is the reason for doing any kind of quantitative analysis. And it can be as simple as a graph, or it can be a very mechanistic model. So it's a large spectrum that we are talking about when we talk about modeling and simulation. So we start with the information that we usually collect in the trial. It can be very early on based on in vivo, in vitro, and you build a quantitative drug disease models and simulations to drive the decisions I talked about. So this is going to be a theme of my presentation. What I'm going to talk about is how we think about leveraging knowledge, or how we think about using the data that we have in regulatory decision making. And there are going to be two perspectives in this talk. One perspective you heard from Dr. Milligan's presentation, that the the drugs are failing in the late phase of drug development. So FDA has already recognized that problem, and critical path initiatives, or most recently, driving medical innovation, the white paper that was published by FDA, really gets to that point as to more investment in this area, which is apparent in this slide. As if you see over the decade, the growth of pharmacometrics at FDA has increased tenfold in the last decade. Beginning of 2000 or 2001, we used to do about 20 odd IND, ND, and BLA reviews. So these are our reviews that we do at FDA. And over the last 10 years, we have gone from about 20 reviews to about 160. And this year, I think we are hitting about 200 mark. So when I joined in 2003, it was 4 or 5 folks. Today, we are at the capacity of 20. So the point I'm trying to make is that the workload has increased, but also FDA has put more investment into getting these expertise at FDA, and also making sure that we do these reviews and have the decisions. What is that we have produced in return? And what is shown here is the pie chart for the reviews we have done in 2007, 2008. So mostly our reviews after the trials is done, and this is talking about only NDA reviews, the key impact decisions that we considered are evidence of effectiveness. That is, we have either provided more insight into the drug, whether it is effective, because there was some questions around whether one trial failed, one trial succeeded, or there was some uncertainty about the results, or we have provided more insights into the dosing, that how we should treat an individual patient or a subgroup, or even the dose that was studied, or safety, where we could have asked more studies. So if you look at the decisions around approved dose, again, I don't have the denominators, but it's just in percentages. If the pharmacometrics review had not been there, or more quantitative analysis, either by the sponsor or FDA, it doesn't really matter who did the analysis, 45% of the time, it would have led to asking a new study that could have been anywhere between 1 to 6 months. 30% of the time, we could have asked for the study that is less than 1 month, and 25% of the studies that could be greater than 6 months. And this is, you can translate to direct savings, because if not having that particular information would definitely lead to asking for new study. Similar for safety, that in certain circumstances, we have elevated the need in 64% of the time for a study that is anywhere between 1 to 6 months, and 18% of the time, studies that are less than 1 month or 6 months. And you can also translate that into a number of patients, because it's not the duration of the study, also how many number of patients would have been needed in these circumstances. So these studies could have been anywhere between 100 to 400 patients in 55% of the time, and less than 100 patients in 30% of the time, and about 400 patients or greater in 15% of the time. So what I'm going to do here is, later in my presentation, is to show you two examples where, in terms of one case, evidence of effectiveness in pediatrics, that how it alleviated the need of studies, and one case for the approved dose, and how FDA used knowledge to come up with a proper dosing that was not available directly or obviously from the empirical data. So all this growth, that 10-year growth and the impact of reviews that I talk about, has also translated, as it was shown by the presenter from EMA, into guidances. There are several guidances that specifically deal with modeling and simulation, population pharmacokinetic guidance, exposure response guidance, but also you find specific details in the evidence of effectiveness, the clinical evidence of effectiveness document, that if whether you need two studies versus one study with a supportive evidence based on PK/PD, or when you have to extend your drug's formulation to a new formulation based on PK matching, how to do that, and drug interaction guidance of using modeling and simulation population pharmacokinetics to do that. And these are more clinical pharmacology guidances, but you also now find with the recent guidances that we are getting more specifics on what our expectations are. So if you look at the hepatitis C guidance, I only copied actually one portion out of it, but there are several portions that deal with how to use modeling and simulations in every phase of drug development for hepatitis C. And I have an example to support that, how this particular document got transpired by some of the work we did at FDA. So it specifically says, and it's only copied, as I said, for phase 1 trials, but we have also talked about phase 2 and phase 3 in the same document, that initially we expect a lot of mechanistic modeling to support dosing. We expect concentration safety because we recognize early on the data are limited. So you have got to make the best out of it. And at least in my personal opinion, in those early stages, mechanistic modeling has the greatest value. And as you generate more data, you can get more and more simpler in terms of your models. Again, diabetes drug development guidance, if you look at it, specifically asked to generate exposure response data in phase 2 because we recognize that's the best data we can use to inform dosing, and it only talks about exposure response either from efficacy or safety side. And we use that consistently in our reviews at the end of phase 2 or phase 2A kind of meetings. And you must also be aware that there are a lot of initiatives specifically in the phase 2A meeting, and this guidance is not mentioned here because that only deals with how to use quantitative analysis in drug development. And this meeting is specifically designed to engage industry and regulatory agencies in conducting modeling and simulation early on so that you can inform the decisions better. And these advices are not binding, so you can talk more freely about some of the model caveats that you have, or the direction you want to go, compared to end of phase 2, which is a little more binding than end of phase 2A. In terms of the scope, as I mentioned, the primary scope is a review, but we also do research to complement these reviews. So our major review responsibilities are IND, NDAs, and BLAs. And the disease model actually nicely fit into that framework because some of the questions that we get from either industry or internal for the regulated decision making cannot be answered based on one data or just one drug. You need to accumulate information across drugs. And the disease model, in my opinion, is one of the best ways to think about how to leverage that information to either make better decisions or to have a different drug development program than you think of. And I'll have an example for hepatitis C as to how we use that disease model in drug development. The other aspects where pharmacometrics has really made an impact is the QT reviews. It is the IRT QT, which is the interdisciplinary review team for QT at FDA, is actually headed by a member of pharmacometrics group. And their concentration QT analysis does play a pivotal role in decision making. And I'll give you a brief example of that too. In terms of the other activities, and I think it's more related to future, we are heavily invested into knowledge management, which I think is one of the keys. And this might be a good opportunity for this crowd to think about how we can think about collaborating on these issues, because as I mentioned, it's not when we develop drugs, one drug cannot inform everything we need to know about the disease. And knowledge management, combining all the data, is really the future if you really want to get serious about using models to inform decisions. And you'll have to preempt some of the analysis that you're going to do by asking some of the questions specific to the disease very early on rather than waiting for data to generate. So again, I have a very few minutes to talk about this, but this is a very broad topic. But I think some of us should start thinking about what are some of the best efforts, what best ways to focus our energy on knowledge management, because this group, I believe, that does modeling, simulation, meta-analysis, can really drive and can really change how drugs are developed and how we make some of the decisions in drug development or even therapeutics. I think is possible in terms of how to manage, if not individual patient, the patient populations. So with that, I'm going to give you two examples. One deal with pediatrics, and the focus of this particular example is not, again, the technical details, but to drive a concept that in pediatric applications at FDA, pharmacometrics plays important role. And I also want you to take this example as a general concept where we can leverage data to alleviate the need for some of the studies that may be impractical. I think the point was made previously, or maybe unethical. And those some of those examples that come to my mind are orphan diseases or biosimilar, where the burden might be higher than necessary. So these applications, the modeling and simulation applications, the regulatory agencies are amenable to those, and we can think about how to use that to best of our knowledge. So I would think that for pediatrics, we are pretty well established in many areas, but with others we are not. But this is again an opportunity to think about how to what are the expectations in some of those areas where trials might be impractical or might be burdensome. So here is a case where we have used this at least for two products. This is a case for topiramate most recently, but the same application was used previously for trileptal in pediatrics. The concept here, as I mentioned, is to see where the trials are impractical or unethical. Here, the monotherapy trials in pediatrics are impractical because you cannot do a placebo-controlled trial. And however, the adjunct therapy trials are usually done in pediatrics, but in adults, you also do adjunct and monotherapy. So what actually the concept that was derived from the review of trileptal a few years ago, which is applied to topiramate, is first you got to check the exposure response after monotherapy and adjunct therapy. And the question you have to ask is the exposure response similar in adults after these two intervention? And if it is, you can reasonably think about extrapolating the adjunct therapy to monotherapy. Again, my intention here is not to get into technical details. There is a lot of work that has gone from Novartis in establishing this particular concept and how to do that particular extrapolation. But this particular aspect was accepted by agency, and at least two products have gone through this particular aspect in extrapolating and coming up with dosing for trilaptal and topiramate for monotherapy application without doing any trial, even, I think I could be wrong on this, but even doing a PK trial. And the product out of it is a simple graph where you can come up with concentrations on y-axis and dose on the x-axis. And this graph, actually different lines are for different weight groups or different age groups, 2 to 10 years, depending on what dose was given to them. And the horizontal lines are the targets that the black line is the target in a dose. Fortunately for topiramate, they also had uh, data in pediatrics, older pediatrics, 6 to 9 years, at a higher dose. So you can also evaluate that target because the safety was deemed appropriate there. So at the end, the label actually reads the black line, which is now the horizontal on the x-axis, as the starting dose for each of the age groups, and the maximum dose for which is the red line for the pediatrics, anywhere between 2 to 10 years. The point I'm trying to make is probably you can do this on back of the paper also in terms of what that dose would be. But this kind of applications give you a very clear picture in multidisciplinary setting to talk about what is the dose, what is the exposure you're going to get. And this is something will be very useful to get from the industry too. And this work was actually done by industry and which was validated at FDA. The second example I want to talk is about the role of modeling and simulation in deriving dosing. And here again, I want to make this example as a very general concept as to use the available information where possible without asking new studies only because regulators could. And the concept here is, it's a very important concept to understand the difference between the evidence of effectiveness and the dosing that goes in the label. It is not necessary for us to write the doses that was exactly studied in the clinical trials. And several of the FDA leadership is very clear on this, that you can post hoc derive doses to best treat patients based on the data, and evidence of effectiveness, which is more a statistically oriented concept as to whether the drug works. Once you establish that, however you do it, using model based or using a simple two-way comparison, the next decision that FDA makes is about dosing, which is the next critical decision in my opinion after approval, can be by using leveraging a lot of data. And there are several examples around that. So I'll just give you one example, which is the case study of boceprevir, the hepatitis C drug that was recently approved. And again, technical details are not important, so I'm going to run through very high level. Merck did 2 studies, Sprint 2 and Respond 2. Sprint 2 study was in untreated hepatitis C patients. Respond 2 was in treated hepatitis C patients. Treated in the sense they were treated with peg interferon ribavirin before, but they failed on it. Well, until these studies were done or these applications came in, there was a thinking in the field that these two populations are different. So what Merck did, or actually back then Schering did, they had different plans in terms of what therapies they evaluated for these in these two studies. So the comparator in yellow, the peg interferon ribavirin, which was the standard of care back then, was the control arm. And the comparator arms, there was one common comparator, which is boceprevir for 44 weeks, which is added on to peg interferon ribavirin, which is also for 44 weeks, which is common in two trials. But there were 2 different approaches. Boceprevir 24 plus PR in a response-guided therapy. Again, it's not important to know what that specifics are in untreated patients, and response-guided therapy with 32 weeks, so difference of additional 8 weeks in treated patients, because as I said, there was some belief that these treated patients are harder to treat. Well, until we did this review, we realized actually these populations are the same. There is a lot of similarities between treated and untreated patients. Again, you'll have to take my word for it why they're similar. But if you think about that, we can combine both trials together. We now suddenly have a lot of data about each of the trials. Well, the second aspect was important in this particular application, that in Respond 2, Merck excluded a specific, which is the whiteout from this particular slide, It was called null responders. They did not prospectively study that. Well, as I said, because we could combine the data, what we did, we actually informed the trial, the population that was excluded in response 2 by using the data from sprint 2. So there was extrapolation from the untreated population to the treated population, and the dosing that was derived and the evidence of effectiveness in the population that was excluded null responders was from the untreated patients. Well, what we also found during the process of the review, that the boceprevir 24 plus PR was suboptimal, which was what sponsor wanted in their label, was suboptimal for some another subgroup in the untreated population. But again, you have to think backwards now, because these patients are similar. We could identify the subgroup from the treated population. So we changed actually the dosing, and here again the option we had was, if not 24, 44 boceprevir 44 could have been adequate. But again, if you just want empirical data, you can write in a label saying, yeah, fine, we found that 24 weeks is suboptimal, 44 weeks is studied prospectively, so that's the dosing. But we also weighed in what is the toxicity that goes in, and is there a different option? And 32 weeks of boceprevir was found to be sufficient. Again, this is, as I mentioned, technical details are not important, but the point I'm trying to make is, from response to the trial that excluded the prior null responders, was informed based on a study that was done in untreated patients, and some of the dosing that was suboptimal in untreated patients was informed by using response to trials. And again, concept here is that when you have this rich data, you can do these extrapolations. But if you have very sparse data, this will be tough. So again, this was not planned, but due credit to Merck and Schering to generate this rich data that allowed us to do this extrapolation. And also, I would mention here that for these two decisions, fortunately, EMA and FDA were on the same page. We had a lot of back and forth with Philip on this, and it's the same decision that was also made at EMA about the dosing in these subpopulations.
So again, just 2 minutes to wrap up, please, Dr. Jadhav.
Yeah. So I will skip next two slides in interest of time, and talk about then why did we do this? We could have just taken a simple stance that you excluded a population, go ahead, study, and give us an empirical data. And also, we found a suboptimal dose, so simply go with the empirical evidence, which is boceprevir 44, which is longer treatment. But I think we need to factor in the public health and the business impact on this. It's not easy to do these studies, especially if you think about HCV, how rapidly it's changing. So the asking studies was impractical, and we looked into the data and derived what is a reasonable dose. Unfortunately, still Merck went ahead and did the trial, probably for marketing reasons. That the study that was just released at the ASLD meeting last month, that they found that the effect size we predicted based on this extrapolation was exactly 38%. The review said 30 to 38%, and the empirical data showed 38%, which is reassuring. But again, from my point of view, even though they did the study, at least the access to market for prior null responders and some rational dosing was given in the trial. And there are other examples which I will not go into details about in the interest of time, but just as a case for Herceptin, where again we found, based on more exploratory analysis, a subgroup that was potentially lacking in benefit. So again, this is done from the regulator public health point of view. So it's not always that it saves you money. It actually asks for more study. But again, if you had used all this information before, you could have planned a better study. And I think the slides will be available, so I will let you read the other examples where modeling and simulation has played a pivotal role. So I'll close this with my key points, as I mentioned, that the demand has increased over the last decade or so at FDA. Our responsibilities they span from review, research, official guidance, and policies. There are several examples which I cannot cover that have made impact on endpoints, analysis, and how the clinical trials are analyzed. And we have played, the quantitative analysis does play a pivotal role in approval and labeling at FDA, and it's only a matter of time, I think. But we also have to um, get clear clarity on the focus as to what the future lies, and that's, in my opinion at least, is the knowledge management and disease model, which I hope we'll be investing in future. So with that, I thank you very much for your attention,